of it all. So here, this is the content we're going to be covering today. Right here, these are the topics we're going to be covering today. We will go through this very carefully, step by step. Do not worry, we have a bit of time. Okay, first part is comparing and contrasting cell types. So with comparing and contrasting cell types, there are technically two, but three you need to look at. So there are main two types, eukaryotic and prokaryotic. So eukaryotic being our animal cells, so the cells in our body, plant cells, and prokaryotic is more bacterial cells. So when you're comparing the two, a lot of the times in questions, they will ask you to write about what's the difference between a eukaryotic cell and a prokaryotic cell, or they ask you to compare an animal cell and a plant cell. And you need to be able to know the structural differences between all of these, because these are very common questions and they usually are 6 markers. So it's very important that you know the differences. It's also important to note them individually rather than just comparing them, because sometimes they do ask you comparisons of not just these 3, but another cell that you may have not heard of. So let's just go through quickly. Eukaryotic cells, generally, they all have nucleuses. They have nuclei, they have mitochondria, and they have ribosomes. And because they all have nuclei, they have the DNA, which is inside the nucleus. They both have cell membranes and cytoplasms, and they're both relatively on the larger side compared to prokaryotic cells. So they would be about 5 micrometers to 100 micrometers. Prokaryotic cells, so prokaryotes, they do not have a nucleus or mitochondria. However, they do also have ribosomes, and because they don't have a nucleus, their DNA is floating just in the cytoplasm. They also have cell membranes inside the cytoplasm, and instead of the DNA that we have in our bodies, like in humans or in animals or in plants, they have circular DNA. It's called a plasmid or a plasmid. And these things, we do not have plasmid or circular DNA. Our DNA is double helix. It's also important to note the differences between animal cells and plant cells. So in animal cells, the only kind of general label diagram that you need to know with animal cells, they usually only need you to label cell membrane, cytoplasm, and nucleus. Very rarely do they ask you to label the mitochondria and ribosome as well. So usually these 3 structures will kind of give you enough. It looks like an egg. Plant cells look very different. They look a lot more complex because they have a lot more elements in them. So they have a cell wall, which gives them mechanical strength. So do prokaryotes, but they're not made of cellulose. So in plant cells, they're made of cellulose. In prokaryotic cells, they're made of something else. I know it tells you what it's made of. You don't actually need to know that a lot of the times, but that's just extra information. In plant cells, they also have chloroplasts and they also have a vacuole. So that's really good in comparing an animal cell and a plant cell. And talking about sizing as well, prokaryotic cells are a lot smaller compared to eukaryotic cells. So that's just comparing the eukaryotic cells of prokaryotic cells, animal cells, plant cells. And if you guys want to work on being able to label them, because sometimes they do ask you to draw a diagram or they ask you to label it, make sure you look at different diagrams so that you're able to identify each structure. Because sometimes looking at the same diagram from your notes, it might just brainwash you to being used to looking at the same diagram. So look at a variety of different ones. Okay, let's move on to the next topic. The next topic is going to be microscopy and magnification. So there are 2 parts to this. There's the theory part of it, which is kind of like the actual microscope itself, light microscopes, electron microscopes. And then we have the second part of it, which is a calculation question. And 9 out of 10 exams, they will ask you a calculation question on magnification. So it's good to point this out. But let's look at the theory part first. First part will be the light microscopes and the electron microscopes. So I believe the ones that you use at school are light microscopes. You need to know that they were light microscopes. They were invented first. They have a maximum resolution of 200 nanometers. And knowing the structure on the side, you can see a diagram. You need to know each of those individual parts of the microscope. So the eyepiece lens, it has a 10 times magnification. So just from looking into the eyepiece lens, that will already give you magnification. Then you have 3 objective lenses with which magnify even more, and that's times 5, times 10, times 25. I don't think you need to know numbers too specifically, but that's just extra information. The electron microscope, they were invented a lot later. And instead of using light, so light microscope, it uses light, as in the name. Electron microscopes, they use a beam of electrons instead. And this means using a beam of electrons, it has a much higher magnification and resolving power. So resolving power meaning resolution. This allows us to have an increased understanding of subcellular structures. So when a question asks you about these microscopes, sometimes they do ask you to compare them. You just need to know light microscopes uses light, electron microscopes uses beam of electrons, and electron microscopes have a much higher magnification and a much higher resolution. You need to mention both, not just magnification, you need to mention resolution as well. And with that, it's also important to note that electron microscopes are a lot more expensive compared to light microscopes. So that's why the school gets light microscopes instead. On the diagram on the right, we'll just quickly walk through it. Just a few of the things. So the stage that holds the specimen, so whatever you're looking at. The specimen essentially is on a microscope slide and it's under a cover slip. You also have the coarse focus and the fine focus. The coarse focus just moves very quickly to move the stage closer so you can see the specimen more closely. And the fine focus moves it very, very finely, very, very slowly so that you can very quick, like slowly adjust to seeing the specimen in a very clear image. Okay. Now let's move on to the math part of it. So I'm gonna give you guys about 1 or 2 minutes to do this question. And I will walk through it with you after giving you a minute or two. Okay, I think you guys probably have had a good look at the question. Let's walk through it. So the biggest mistake that students make when they're doing magnification calculations is the units. This is because the calculation itself is actually quite self-explanatory. It's just one equation and you just need to rearrange it. The biggest mistake is not looking at the units very carefully. So let's just have a look at this question. Okay. Right. When you're doing these magnification calculations, you already get a mark for just writing the equation out. So I just recommend writing the equation. So this would be, in this case, image size over actual size gives you the magnification. So I over A equals M, or however you want to memorize it. Rearranging the actual equation also gives you another mark. So just by doing that, you're going to get 2 marks in the question already. So it's important to you write the equation and then you rearrange it. So don't be lazy and just put in the numbers because sometimes they might not give you marks for that. They usually do, but it's just safer to write this down. Then you're going to put in the values. So in this question, it tells you that the length of the cell that you're seeing is 4.8 centimeters, and the magnification is 400. So you just substitute the values, rearrange it, and you see that actual size equals image size over magnification. So 4.8 over 400, that gives you 0.012 centimeters. Now, here in the question, it is asking to give your answer in micrometers. So you have to be able to convert centimeters to micrometers, which in this case is 120. So just have to emphasize this again. It is so extremely important that you write down the correct units. So make sure you're converting them correctly and making sure that you're actually reading the question properly. So be very, very careful. but this is just how you break down the question. If you have trouble with math questions generally, because there's a lot of math and sciences in general, especially physics, do as many as you can and try to remember as many of the equations as you can. So with this question, there's not much I can really break down other than be careful with units and do more math questions. Okay, let's move on. Transport through membranes. This topic is so extremely important. This one is such a massive topic and it comes up every exam. I can't guarantee that for sure, but it comes up almost every single year in GCSE and you need to know it well. So let's break it down. There's diffusion, osmosis, and active transport. Diffusion is, you need to know all the definitions by heart. So diffusion, defining it, is net movement of particles in solution or a gas form from an area of a high concentration to an area of a lower concentration. So from high to low, essentially. There are factors that affect diffusion rate that you also do need to know because sometimes they ask you to apply it to the question. So here we talk about steepness of concentration gradient. So imagine you have, let's say, a bunch of particles going around and there's a membrane. There is, let's say, 80% of particles on the right and like 20% of particles on the left. That's going to move very quickly and have a very fast diffusion rate because there's such a steep concentration gradient. So if there were only 30% of particles on the right and 20 on the left, it's not going to be as quick because it's not as steep. So the steeper the concentration gradient, the faster the diffusion rate. Temperature works similarly. The higher the temperature, the faster the diffusion rate, higher the diffusion rate, because higher temperature, more energy. Surface area, similar again. The larger the surface area, the higher the diffusion rate. Diffusion distance is the opposite. The shorter the diffusion distance, the higher the diffusion rate. And the reasoning, look at the diagram in the bottom left corner, you can see that there are the capillaries. And it is the alveoli, and it's just doing gas exchange. The capillaries are 1 cell thick. They are very thin. And because think about if you're moving stuff from very thin, diffusing stuff, like moving stuff over, it's going to be a lot easier than transporting very thick layer. Think of it like a very thin towel. If you have a very thin towel and you pour water over it, more water is going to come through if it's thinner. If it's very thick, there's not going to be as much water going through. So that's kind of how you would think about when it comes to diffusion distance. So the shorter the diffusion distance, the higher the diffusion rate. One thing I want to point out for those who are doing AQA, you do actually need to know more specifics on diffusion. So things like intestinal villi or alveoli, you need to know in detail on what the adaptations are. So I'll just quickly go through that with you. If it's alveoli, capillaries, or whatever, they are 1 cell thick. They're very thin, so short diffusion distance. Alveoli do have a very large surface area. Same with villi, they have a very large surface area. And they also, for villi, they have a lot of microvilli, increase the surface area. That's just kind of what you need to know for AQA specifically, but generally everyone else does need to know the factors affecting diffusion rate. This goes with everyone else. Right. Let's move on to osmosis. This is where people get a little confused because they're very similar. Osmosis is a type of diffusion, and osmosis is movement of water. So if you think about it, the osmosis is diffusion of water from a high water concentration to a low water concentration. A high water concentration meaning a dilute solution, and a low water concentration meaning a concentrated solution. So if you were to write the definition of osmosis, it would be the diffusion of water from a dilute solution to a concentrated solution. And that's one part of it. Second mark, people always forget, you have to write this down, is through a partially permeable membrane. When you're talking about osmosis, you have to talk about through a partially permeable membrane. Okay, there's an experiment that you guys might have done for osmosis, but I'm not going to go through the details of the experiment, but I will talk about like the application of it. So osmosis, usually cells can be put in solutions with different relative concentrations. And there are 3 things you need to know with those concentrations and the relative concentrations. First one is isotonic, where the cell is put into a solution that has the same concentration as the cell. Isotonic. There's no movement because the concentration is the same inside the cell and outside the cell. This is not written on here, but just to tell you the term, just because sometimes they do use that term isotonic. Same with hypertonic, hypotonic, they will give you these terms. Hypertonic, things like a salty brine. So a very, very, very concentrated solution. So it's more concentrated outside the cell than inside the cell. Hypotonic is the opposite. It is when it is very, very dilute, things like pure water. So it's going to be more concentrated inside the cell than outside the cell. Okay, I'm going to walk through why you need to know this. In a hypertonic solution, where it's very, very concentrated in the very, very concentrated solution, and the cell is not as concentrated as the solution, because there's so much water inside the cell and it's less concentrated, so more water in the cell, water is going to want to move out of the cell through osmosis. Through this, because there's water moving out, cells shrink. In animal cells, we use the word shrunken or shriveled. For plant cells, we use the word plasmalized. So it starts flaccid and it becomes plasmalized. It shrunk. Hypotonic is when it is the opposite, because it's things like pure water. It's going to be more dilute on the outside. So water is going to move from the solution into the cell through osmosis because there's more water outside. This is going to cause the animal cells to swell up. And sometimes, because there's so much water going in, it's going to burst. In plant cells, we use the term turgid for describing cells that are filled up with a lot of water, essentially. However, it's good to think about why plant cells don't burst, because plant cells have a cell wall. They have mechanical strength, therefore they do not burst compared to animal cells that will easily burst because there's so much water going in. They don't have a cell wall. So that's something to think about. But you do need to know the three different types. Isotonic, same concentration. Hypertonic, solution is more concentrated. Hypotonic, solution is more dilute. Okay, let's move on to active transport. With active transport, the definition is basically the exact opposite of diffusion. It's the movement from a dilute solution to a more concentrated solution. So not dilute solution of water. So it's a movement from a less concentrated to a more concentrated solution. So it's from low to high. Diffusion is high to low. Active transport is low to high. And this is the first mark when you're defining active transport. Second mark, you have to talk about how it requires energy. In the name itself, active transport, it is active. It's an active process. Therefore, you need energy for any movement. And this energy comes from respiration. So the examples they use a lot, and I would say that you probably do need to know, is two things. Absorption of sugar molecules. So usually sugar goes from a low concentration in the small intestines to a higher concentration in the blood. Second one will be the absorption of mineral ions. Usually there's a very, very little mineral ions in the soil. And because plants need those mineral ions, they're going to go from a low concentration in the soil up into the plant root hairs. Plant root hairs, they have a large surface area that allow them to absorb these mineral ions very, very easily. So that's just one adaptation. But essentially, it goes from a low to high concentration into the plant root hairs. Okay. That is transport through membranes. We're going to quickly look at a question here. This applies more to AQA, if anything. As I've said, AQA does ask you to look at the alveoli and villi, and that you need to know the adaptations. So I'll just work through this really quickly with you. This is AQA, you need to memorize it, but everyone else who does all the other exam boards, you still need to know that if they tell you there's a large surface area here, you need to be able to figure out, oh, the higher diffusion rate, more absorption. So in this question, it's asking you how the alveoli and the villi are adapted to increase absorption. A question itself, you just need to talk about how they have a large surface area, which allows it to maximize diffusion. So villi and alveoli both have very large surface areas. Especially with villi, they have these microvilli. They also have very short diffusion distance, a very short diffusion distance because they have very, very thin walls. So again, thin walls, short diffusion distance, higher diffusion rate, better absorption. Something that I feel like is also good to note that they don't mention within diffusion that applies specifically to alveoli and villi, so our biological structures. They have very good blood supply, or I would like to say capillary network. So they have a very good capillary network allowing them to maintain a very good concentration gradient. An extra bit of information as well that is actually good for application in biology is that villi, they have a lot of mitochondria because they have a lot of mitochondria that allows them to have a lot of energy. So for active transport. So that's transport through membranes. Just it's important to note all of that essentially. I don't think there's any more that you need to know within transport through membranes when it comes to content. It's more about knowing how to apply your knowledge to the questions. These are very application heavy, so know your basics. Okay. Before I move on, let's just see if you guys have any questions regarding that. What type of cell is a prokaryotic cell? The prokaryotic cell are things like bacteria. So that's like the most basic one. You usually probably only get bacteria. So you probably have like a round kind of oval-ish looking thing with a little tail, the flagella. So that is kind of what a prokaryotic cell is. I don't think you need to know the other types, but it's mainly just bacteria. I think that's kind of all you need to know. Okay. Wait. Let's see. What is the max resolution of an electron microscope? The max resolution is like very small. Wait, hold up. The max resolution is 0.002 micrometers. You don't need to know that, I don't think, in specific, but if you want to know that, I think that is the answer. Yeah, I think that is kind of it for that. Fick's law. Oh, I think that is dependent of which exam board you do. I don't think this is for everyone. For Fick's law, I think you just need to know the equation, but generally it's just movement of particles from a high to a low concentration, and knowing that it go from a high to a low concentration. So as I've said, diffusion, moving from a high to a low concentration, that theory is directly proportional to the particle's concentration gradient. That's the best way I can explain it. I don't think you need to know further than that theory. Besides, you need to know Fick's law is just diffusion. The diffusion flux, essentially movement of particles, is directly proportional to the concentration gradient of the particles. And you have this equation, and you might need to apply this equation. That's the best way I can explain it, but I don't think you need to know that in too much detail, so don't stress too much about that. Okay. Is it okay if things are slightly slower for note taking? Yeah, that's completely fine. I'll try and slow down slightly. You do have the slides in the chat. So if a lot of the times I am reading some of the notes on thing, just try to, if you want to note something down, maybe note what I'm saying that is not on the slides. So that you can kind of like catch up, because I'm trying to go as like quickly as possible to go through all the content. So note down what I'm saying that's not on the slides, but I'll just note down everything, but I'll try and slow down. Okay. So for the questions for now, let's move on to the circulatory system. Right. The circulatory system is very, very complicated, is one of the biggest things we have to look at, because it's so complicated. The heart is so complicated, and you guys need to know a lot about it. So we'll work through it slowly. The heart anatomy, I'm not going to explain much on that. You just need to know kind of the labeling of it. As I've said before, with the heart and any structure, essentially, you need to be able to label them. And by doing so, you should look at different types of diagrams. So go on Google and search structure of heart, diagram of heart. Look through different pictures and see if you can label all of it. Because looking at the same, so if you look at these slides and you look at the same picture, you're not going to be able to label it when you see a different diagram in the exam. It's very difficult. So it's great that if you can look at different diagrams throughout. So main parts of it, I mean, it's all labeled on the left there. I don't think I have to talk more on that. The heart, the function of the heart, we will go through step by step. It is a bit more complicated than that. Sorry. Is this in combined higher? Yes, I believe so. Let me double check. Yes, this is in combined higher. It is in combined higher, yes. So with the heart function, let's just go through this. We're going to talk about how blood flows throughout the heart. and we're going to talk about deoxygenated blood and oxygenated blood. So first up, the right ventricle is going to pump deoxygenated blood, so blood that doesn't have any oxygen, to the lungs for gas exchange. So think about it. If you want to get some oxygen into your blood, you're going to pump some deoxygenated blood to the lungs to get this oxygen. So the right ventricle pumping deoxygenated blood to the lungs to get oxygen for gas exchange, removing carbon dioxide, taking in oxygen. Oxygen enters the blood, carbon dioxide leaves the blood. And this is thinking about you're breathing in, you're breathing in oxygen, going into the lungs, the oxygen, transport all this oxygen into your blood. Carbon dioxide leaves your blood back to the lungs. And when you breathe out, you're breathing out carbon dioxide. The left ventricle, it pumps oxygenated blood. So this is the other ventricle. Be very careful with this. The left ventricle pumps oxygenated blood around the rest of the body. And why it does that is so that it can provide the rest of the body oxygen for aerobic respiration. The rest of the body uses a lot of energy. Things like running, exercise. You're going to need oxygen in your muscles. So, and also even in your heart, you just need general oxygen. So the left ventricle pumps all of this oxygenated blood that you've taken in from the lungs and pump it all around your body, to your brain, to your muscles, to your legs, everywhere, literally everywhere. And this allows for aerobic respiration and energy. That is the kind of the pathway of blood alongside with the heart. For valves, so you can see on the left, you have valves between the atrium and the ventricles. The valves, the point of it is to stop the backflow of blood. So it stops blood from going the incorrect direction. Pacemaker cells. So these pacemaker cells, they're located in the wall of the right atrium. So this is something you need to know kind of for applications. They ask you about pacemaker cells a lot of the time, and I'll explain why in a bit. You'll see why. So you need to know that these cells are located in the wall of the right atrium, and they control heart rate. So these pacemaker cells help the heart rate be regular. Irregularities in the heart rate, so sometimes there might be dysfunction in the pacemaker cells. Something's wrong with the pacemaker cells. This causes irregularities in your heart rate. So it's not pumping at a regular time. So this is corrected using what we would call an artificial pacemaker. They're electrical devices. And these artificial pacemakers help regulate the heartbeat to a normal heart rate. So that's why you need to know about the pacemaker cells. Pacemaker cells maybe go wrong, then there's irregularities in the heart rate. Artificial pacemakers help make it go back to normal. Okay, the coronary arteries. So this is not labeled on the diagram. I think this was very tricky. I never knew you needed to know where it was, but in my GCSE, I was asked where the coronary arteries were. So look at different diagrams. This is what I mean. Coronary arteries, they deliver oxygen for aerobic respiration. That is their function, their main function, and kind of the main artery that kind of delivers oxygen. And coronary heart disease comes up a lot. Coronary heart disease can lead to a heart attack. And the reason is because there is fatty material called these plaque building up in the coronary arteries. So think about your arteries as like a pipe. So like a pipe. When you have coronary heart disease, it's like sewage being built up in these pipes. Water is not going to be able to flow through these pipes as easily if there's a bunch of sewage built up. That's similar to how coronary heart disease works. There's a pipe, so your blood vessels, fatty material, so plaque, builds up. Less blood can flow through. If less blood can flow through, there's less oxygen going to your heart muscles. That causes your heart muscles to die because without any oxygen going to your heart muscles, no respiration, no energy, it just dies. So without oxygen to the heart muscles, they die. This can cause a heart attack. So this is kind of like the kind of mechanism, the step of actions, the steps to what happens in a heart attack is because of your a heart disease, like coronary heart disease built up. That's kind of the explanation of it. They might ask you an application question on this. Something I want to point out very carefully, though, is it's important to know that the blood goes to your heart. It goes to your heart. You have oxygen around your heart for respiration, to give it energy, and then it goes all around the body for energy. So it goes oxygen you get oxygen from the lungs, then to the heart heart brings it to everywhere else in the body, brain, muscle, all of that. Okay. Before I move on, let's go through because this is quite a difficult one. So I'm going to see if you guys have any questions regarding that as well. This slides are I don't the slides are not on the chat. They should be. Ooh. They should be sent to you. Okay, they will be sent to you at some point, but they definitely will be sent to you. Don't worry about that. Will be yeah, they will definitely be sent to you, so don't worry. We will send them to you. Okay, how does aerobic respiration provide energy? Aerobic respiration, we will go through respiration in a bit actually, but just to give you a very brief understanding of aerobic respiration. Aerobic respiration, you take in oxygen and then you with glucose in your body essentially, and that is kind of broken down. That produces ATP, which is a molecule, and ATP kind of breaks down to provide energy. You don't need to know this in too much detail, but essentially ATP with aerobic respiration, it produces ATP, and ATP gives you energy. That's kind of all you need to know. And you don't need to know too much detail in it. So aerobic respiration gives you energy. That statement itself is enough for GCSE. Okay. What's the septum? The septum, I don't know if you can see my mouse. The septum is just the end of the heart. So you know that little tip at the end of the heart there, that's just the septum. When it comes to structure, it's not really the function of the septum is just to separate the right ventricle and the left ventricle. It's like a wall that separates the two ventricles. That's kind of all you need to know of what the septum is. I hope that answers your question. But yeah, a wall separating the two ventricles, separating the chambers of the heart. That's kind of all you need to know about the septum. This depends on which exam board. Some exam boards don't even mention the septum. But you know, that's that. Okay, let's move on. Let's talk about vessels and let's talk about the blood. And then we'll go an exam question because the exam questions are quite difficult, I will say. Okay. A lot of the times for the circularity system, when they ask you about blood vessels, they always ask you about comparing these vessels because they're very different. They look similar, but actually quite different structurally. So let's just talk about the differences in their structures and their functions. So first, let's talk about the artery. It transports oxygenated blood away from the heart at a very high pressure. So I'm going to slow down with this because this is not written on the slides, but I will talk about this. So artery, I'll reiterate, it transports oxygenated blood away from the heart at a high pressure, a very, very high pressure. High pressure is the main point. You need to highlight that, high pressure. Okay. It has a very thick outer protective wall. So you can see on the diagram there, it shows you there's a very thick outer wall. And it has a very thick muscle and elastic layer, again, written on the diagram. And the reason for the thick muscle and elastic layer is so it can withstand this high pressure. So as I've said, this transports at a very, very high pressure for the artery. And how it deals with this high pressure is through this thick muscle and elastic layer. It has a very small lumen. So the lumen is essentially like the pipe that allows the blood to flow through. So yeah, with basically the hole, the pipe, I think that's the best way I can explain it, the pipe that allows the blood to flow through. The small lumen also helps to maintain high pressure. And this is also not written on there, but they have a very smooth lining to minimize friction. So I'll let you guys maybe like 30 seconds to write this down. There's some information that's not on there. So, but yeah, smooth lining to minimize friction within the arteries. Okay. Vein, the next one, let's move on to the next one. The vein, it transports deoxygenated blood. So the artery transports oxygenated blood. Vein transports deoxygenated blood back to the heart and is at a much lower pressure, a low pressure. So, yeah, I'll reiterate. So to let you guys write this down if you want to know this down. It transports deoxygenated blood back to the heart. So for the artery, it was oxygenated blood away from the heart. Vein is transporting deoxygenated blood back to the heart. And this is at a low pressure. Something I want to point out that I didn't point out earlier is that the pulmonary artery is an exception and the pulmonary vein is an exception. So the pulmonary vein, it carries oxygenated blood from the lungs. So that's the only exception. The pulmonary artery, pulmonary vein carries oxygenated blood from the lungs. That's the only exception to what I've said earlier. So something to just note, to be careful of. So it has a thinner outer wall, as seen on the diagram, and have a thin layer of muscle and elastic, again, also written on the diagram. Large lumen, and it also, similar to the artery, has a smooth lining to minimize friction. There is one thing that stands the vein out compared to the artery and the capillary, and that it has valves. This is very important to note. It has valves. So this is not shown in the diagram, but the valves are usually like little, I don't know how to explain it, but little, how to explain that? Like a little, looks like this. I don't know if you guys can see my face, but it looks like that. And it's in the lumen, basically. And that's called a valve. Usually you will be able to see in the diagram, because usually with like the artery and the capillary, you see it's just like a very clear hole. But with the vein, there is something on top, and within the lumen. And that is to make sure that blood flows one direction. There's no backflow. So that's very important to note down. Valves make sure there's no backflow of blood and ensures that blood flows one way. So that is the vein. Right. With capillaries, I think capillaries. Okay. With capillaries, this is also extremely different compared to artery and vein. So the artery and vein, they look somewhat similar. The capillary, even though in the picture it doesn't look like it, it is much, much, much smaller compared to the vein and the arteries. So capillaries, what they do is they surround respiring cells. So this is something you need to note down. They surround respiring cells. And they transport blood at a lower pressure, a very, very low pressure, like much, much lower compared to artery and vein. Very low pressure. I'm going to reiterate. Capillaries surround respiring cells, and they transport blood at a very low pressure. The reason for this is because it allows time for gas exchange. So the main thing with capillaries is that, so think about capillaries, they're usually around the alveoli. Around the alveoli, that allows for gas exchange. And it is, because it's moving so slowly, it allows time for gas exchange. They have a very small lumen, as seen there. And they have a very thin wall, as I've said, and this allows for short diffusion distance. This was mentioned a few times before. Something extra, I think, depending on the exam board, you need to know this. Blood plasma, they do pass through these capillary walls into the tissues. And when these blood plasma, I'm going to reiterate again, blood plasma passes through capillary walls into the tissues, this is what we would call a tissue fluid. I'll say this again. Blood plasma passes through these capillary walls and into the tissues. And this is what you call tissue fluid. This, again, heavily depends on what exam board you do. I don't know if every exam board needs to know this, but this is something to point out. Okay. We're going to move on. This is, I think, all you need to know with vessels, just comparing and contrasting them. I've just mentioned all the points you need to know. If you missed anything and you want me to reiterate, please put it in the Q&A. I will read it in a bit, but I want to go through blood as of right now. So let's go through the next section. Okay, this is kind of the structure of blood. there are four parts to the structure of blood. There's the plasma, the red blood cells, the white blood cells, and the platelets. So with the blood, the plasma, I'll talk about the structure and function of each of these. Plasma is essentially what makes it liquid. It's the straw-colored aqueous liquid. You don't need to know that that's a very nice wording of it. It's just the liquid in it, what makes the blood liquidy. In the plasma, it has other blood components in the plasma. So things like it transports glucose, amino acids, carbon dioxide, and urea. I'm going to go through this a bit more quickly because this is all written on the slide. But if I say any extra information, note that down. Red blood cells. So they contain hemoglobin, and the hemoglobin is what binds to the oxygen. That's important to know. So the red blood cells, there's a much more complex structure in the red blood cells, but you don't need to know anything in detail. The only thing you need to know within the red blood cells is they have hemoglobin, and that's what binds to the oxygen. Their shape is biconcave, and this allows it to have a high surface area to volume ratio. The main function of it is that it transports oxygen around the blood. So because it's binding to the oxygen, that's what is kind of binding onto it and then releasing it and allowing us to respiration. That's red blood cells. With white blood cells, there are two types of white blood cells you need to know. There's the phagocytes and there's the lymphocytes. I know some of you guys do infection and response, and you need to know a bit of detail on like how the phagocytes kind of attack these cells, but I don't, because this is not in every exam board, I don't want to go into too much detail of it, but I will explain briefly. So sometimes they might ask you to identify these lymphocytes and phagocytes. Oh, the light just turned off. Okay, anyway, the phagocytes have a lobed nucleus and a granulated cytoplasm. So that's how you'll be able to kind of differentiate between phagocytes and lymphocytes. So the phagocytes, it will kind of look like as seen in the diagram below. So the white blood cells, they are part of the immune system. This is what protects your immune system. Essentially, they help you destroy pathogens. Pathogens are what make you kind of sick. They're like toxic, they're little, think of it like toxic little creatures trying to make you sick. That's what pathogens are. So the phagocytes, they engulf these pathogens. What engulf means, they kind of just eat them. Don't use the word eat. They engulf them. And once they engulf them, they digest them using the enzymes. So as I said, there's like the cytoplasm is granulated. These granules, they have enzymes. So these enzymes break down these pathogens. The lymphocytes, they produce antibodies. So the antibodies are something that also helps attack these pathogens. So the lymphocytes produce antibodies and they clump pathogens together. So that's white blood cells, part of the immune system. Platelets, okay. The details, again, I'm going to have to emphasize this. Details depends on exam board, but I'm going to mention basically all the details just in case. Cell fragments is something that are within the platelets. They have these chemicals and they are called clotting factors. I'll explain why this is important later. So they have these clotting factors in the platelets. So when your skin is wounded, for example, you got a cut on your arm, let's say, it's going to start bleeding. So what happens is these platelets release these clotting factors and they turn fibrinogen in the plasma into fibrin. I'm going to explain that. They release clotting factors, which allow fibrinogen into fibrin. This then forms a mesh that sticks these platelets together to then form a scab. This is why you stop bleeding. They form a scab, a protective layer, so you're not just bleeding all the time. That's what platelets does. So I hope that is clear. I'm going to look at the Q&A really quickly. If you guys have any questions on this, please type it right now so I will be able to explain it. If I need to repeat anything, I will repeat it. What is the lumen? The lumen is, okay. Think of a pipe. The lumen is the hole, essentially, how the blood flows through. So like kind of like a little pipe like this, like it will let blood flow through. Oh my God, why is the light off? So if I have a piece of paper, actually, that'll work. Think of this book right here. Think of this as like the blood vessel. I don't know if you can see my face. Think of this as the blood vessel. This is the blood vessel, and the lumen is what's going through in here. This is the lumen, the hole here. This is the lumen. If that's my best way to explain it. What board did I do? I did AQA. But I also taught other students doing like Edexcel or OCR, so I'm trying to explain kind of generally. Somebody just said respiring cells. Can someone elaborate on what that means? Respiring cells, like what do you mean? What do you want me to explain with that? What do the capillaries usually surround? Okay, the capillaries usually surround villi. So it's in your intestines. So they usually surround villi. Main one that they usually ask is the alveoli. So the alveoli, which is the stuff in your lungs, that's also what capillaries usually surround. And with the phagocytes, sorry. With the phagocytes, what did I say about the phagocytes? They engulf pathogens. So they kind of don't use the word eat. I'm only using that to explain things, but they eat the pathogens and they digest them. So that's my answer to that. Okay. Let's look at an exam question. This is quite difficult, I will say. So take your time. I'm going to give you guys about 2 to 3 minutes. I'm going to give you guys actually a bit more time. I'm going to give you about like 2 to 3 minutes to answer both of these questions. And I will come back and explain them. I just need to fix the lights, but I try and do this question. It is difficult, so don't worry if you don't know how to do it. Okay, really quickly before I'll let you guys continue writing just to answer the question somebody asked. Do we have to mention alveoli's large surface area? Depends on the question. So if they're asking you like how it increases absorption or like how it increases gas exchange, is there something on alveoli that is adapted to gas exchange? Yes, you're going to answer because it has a large surface area. However, generally also if they ask you about structure, like oh, what's the structure of alveoli? I guess you can say it has a large surface area. But generally, if it's asking you things like, if it's asking you about blood, and the capillary network, you don't have to mention the surface area. But if it's asking you about structure of alveoli or how it's adapted to gas exchange, like diffusion or anything like that, yes, you do have to mention the alveoli as a large surface area. And the better wording would be large surface area to volume ratio, because technically, if you think about it, its surface area isn't big. It's more compared to its volume that it's massive. That's why it's adapted to gas exchange. But large surface area is fine. You can say that. It's just better to say large surface area to volume ratio. That's my answer to that. Okay. let's just explain that. Let's just quickly go through this question, right? First question, relatively simple and straightforward, I really hope, because we did go through this, explaining the differences between the artery and the vein, as seen on the diagram right there. When you're answering these questions, I mean, I don't think I have to explain the answer here. I mean, it's just telling you here that like thicker muscle tissue, thicker elastic layer, narrow lumen, all of this. Yeah, that's fine. This is all like something that we've explained earlier. It's more straightforward using your knowledge. However, when you're answering comparing questions, you need to use comparison words. What I mean by comparison words, but on the other hand, or in this case, just even ER, so thicker, narrower. Don't say, oh, artery has thick tissue. Or artery has thick elastic layer. You can't say that because then you're not really comparing it. You have to say it has a thicker elastic layer or a thicker muscle tissue. That's just something to point out. The question I do want to look at mainly is the 6 marker on the right. Explain the effect of a partly blocked coronary artery on the human body. So with this question, I will say it is extremely difficult. Even I found it a slightly difficult to do, but we'll go through it. So when it's asking you about a partly blocked, and it's talking about explain, so it's talking about a partly blocked coronary artery on the human body, how it affects the whole human body. So you can't just talk about the heart. You have to talk about basically everything else that's happening. So we have a partly blocked coronary artery. As I've said, there's usually plaque built up in these coronary arteries, therefore less blood flow. So the basics of maybe the first 2 marks you'll get is saying there's a blocked coronary artery, less blood flow to the heart, so less oxygen going to the heart and less glucose going to the heart. There's less oxygen, less respiration, you know? Next part, you talk about less respiration of the heart. Because there's less respiration, there's more anaerobic respiration and less energy. Less respiration means less energy. Well, when I'm saying respiration, aerobic respiration. So that only really earns you about 3 to 4 marks. The difficult part of this question is actually elaborating more and applying your knowledge. So in this case, would be talking about what happens further to the rest of your body. Because we're only just talking about the heart right now. We're talking about how the heart, there's less oxygen, there's less glucose, there's less respiration, and there's less energy. We need to talk about the rest of the body. And because we know that with blood flow, it's going through the rest of the body is receiving less oxygen. Because there's less oxygen around the whole body, and there's also less going maybe to the lungs. There's also less muscle contraction. Think about all of this. And also I said, because there's less aerobic respiration, there's going to be more anaerobic respiration because there's lack of oxygen. So they carry anaerobic respiration. Anaerobic respiration produces lactic acid. Lactic acid causes muscle fatigue. This is a very difficult question. I am very aware. Here is the answer to all of this. But essentially, this question, you know the knowledge. You know that with a blocked coronary artery, there's less blood flow, less oxygen, less energy. It's just thinking deeper, thinking about what happens with muscle contraction, thinking about what happens with the rest of the body. So this is how you have to earn all marks. But I don't want to explain too much like on the content, but more so how we answer this type of question. With 6 markers in biology, a lot of the times, unless they're talking about tests and describing, but even then, they go based off of levels, level 1, 2, and 3. And instead of answering point by point, they want you to answer, so like, even if you answer 6 points from this mark scheme, you might not get all 6 marks purely just because they want you to answer points from each section. They want you to explain everything. So just be careful when you're answering these 6 markers. It is tough, I understand, but after the session, go through this question again, have a look and see whether you can answer it fully with 6 marks. And if this is difficult for you, do more questions because this is more application. And I get it's tough. The next part of it, the next section, I kind of is another example question, but I want to just go through this with you because this is all content-based. First part is asking you about red blood cells, and they talk about comparing red blood cells with the structure of plant cell. So this goes all the way back to when we first talk about comparing and contrasting cell types. So let's think about the plant cell. What do they have? Vacuole, chloroplasts, cell wall, cell membrane, cytoplasm, all of that. 6 marks, you're just comparing the red blood cells. You can say that red blood cells don't have a cell wall. They do not have a vacuole. They don't have a nucleus. Just saying that already gives you 1 mark each. However, I want to also heavily emphasize with comparison questions, you can't just write the differences. You have to write the similarities as well. So if you just wrote differences in this question, you probably will get 5 marks or 4 marks max. You need to talk about the similarities as well. Just wanted to emphasize that. The bottom question is the one I kind of want to focus on more. I'm going to give you guys like a quick minute to read through the bottom question and have a think about it. Okay, I think most of you probably have read the question. Okay, it's talking about the symptoms and how a low number of blood components can cause all of this. So to do this question, we might want to reverse and think back on what blood components we have. We have the plasma, we have the white blood cells, the red blood cells, and we have the platelets. Which of these components match these symptoms if they're low? So I think the easiest one will be bleeding that will not stop after the skin is cut. There's only one answer to this, and that's platelets, because platelets are the only ones that basically form a scab. So you just need to be able to figure out, oh, what is the function of each of these blood components, and then apply it. So bleeding that will not stop after the cut, you just say platelets. If there's a low number of platelets, no clotting factors, they don't form a scab, therefore bleeding will not stop. Frequent infections. What protects our immune system? That's the first thing I'll think of. Infections, right? Pathogens. White blood cells. White blood cells protect our immune system. They engulf these phagocytes. Therefore, there's going to be more infections if there are less phagocytes and less white blood cells in our system. Tiredness is slightly more difficult because tiredness is you need to think a little bit further. So now that I've answered the bottom two, there are 2 left, plasma and red blood cells. I know for sure it's probably not plasma because plasma is kind of one of the, I won't want to say irrelevant, but it is not the most important like component of the blood. So now I'm thinking about red blood cells. Red blood cells, what do? They transport oxygen around the body. And we know from oxygen that allows us to do respiration and give us energy. So therefore, if a person has a low red blood cell count, they're going to have less oxygen transporting throughout the body, less respiration, less energy, therefore being tired. So that will be kind of how you break down this question, this 6 marker. So here is kind of the mark scheme of it all. Okay, I think that's kind of it for now for this circularity system. Any questions on this, please put it in the Q&A now. Yeah, I'll give you maybe like 30 seconds to see if you guys have any questions. But I think I should have answered them throughout, I believe. So should be fine. Okay. We will have a wait, there is a thank you. Right, we're gonna move on. We're currently at like about an hour and 50 minutes in. I want to give you guys about like let's say I'll give you guys a 10-minute break and then we'll continue the rest of biology. I'm gonna stay here and answer any questions that are on there, but I'll give you guys like a 10-minute break. So here's a 10-minute break. Do whatever, have a little break, but I'll look at the questions as we go through. Can you talk about what happens with anaerobic respiration? Okay, I will answer the questions after the break. So type in your questions if you have any, and I'll answer them right after the break. Okay. Break is over. We're gonna continue on. I know somebody asked about anaerobic respiration. We are about to go on photosynthesis and anaerobic respiration, so I will explain all of this in detail in a bit. So don't worry. Let's move on. Photosynthesis. Okay, with photosynthesis, just quickly reminding you, because this is in chemistry, what is endothermic and exothermic? Endothermic means you're taking in energy, and exothermic means you're releasing energy. So photosynthesis is an endothermic reaction because it's taking in light energy. So this light energy from the environment, but from the sun, and this light energy is then transferred into chloroplasts. So within the chloroplasts, we have these things called chlorophyll. And the chlorophyll absorbs this light energy. And after it absorbs it, it's going to produce glucose, which also has many uses. So for example, plant respiration, they make their own glucose. So therefore, it's unlike humans where we actually have to eat to get this glucose. For plants, they make their own glucose through photosynthesis, so they don't actually need to eat anything. The word equation and the symbol equation, I think you guys do need to know. So just a reminder, the word equation for photosynthesis is carbon dioxide plus water through light. That's important. Remember to write arrow and then light on top. Gives you glucose and oxygen. The symbol equation is actually incorrect. Hold on. Wait, give me a second, guys. Oh. Let me just quickly edit. I can't edit that. Wait. Sorry, hold on. Something up with the slides. What do I show you? Okay, should be fine. So anyway, the symbol equation is wrong. I'm so sorry. It's 6CO2 plus 6 H2O light, C6H12O6 plus 6O2. It's not balanced. It's meant to be 6 and 6 on the side for CO2 and H2O. My bad. That's my mistake, but should generally be fine. Then, okay, let's talk about the rate of photosynthesis as well. So because we know that oxygen is being produced and carbon dioxide is also being absorbed, to figure out the rate of photosynthesis, we can figure out by finding the volume of oxygen produced or the volume of carbon dioxide absorbed over time. So that would be how you figure out the rate of photosynthesis. This is the basics of photosynthesis. There are so many questions on photosynthesis and respiration that we will look at in a bit. I'm just going to talk about also the limiting factors of photosynthesis in a bit. So first, let's look at temperature. So molecules, if they have a higher temperature, they're going to have more kinetic energy. This means that they'll collide more often and they're going to react a lot quicker. This causes rate of photosynthesis to increase. However, it's so important to note that when temperature is too high, this can cause enzymes to denature. Denature meaning their active site changes, and like they change shape and they can no longer bind. So when temperatures are too high, enzymes denature and therefore they no longer work. So temperature increases the kinetic energy and increases rate of photosynthesis to a certain point. And when it is too high and the temperature is too high and the enzyme denatures, it's just going to go straight down. There's no more activity because once it's gone, it's gone. Light intensity, very straightforward. Higher light intensity, higher rate of photosynthesis. Same with carbon dioxide. Higher carbon dioxide concentration, higher rate of photosynthesis, because there's going to be more. You're going to need more carbon dioxide. Amount of chlorophyll as well. If there's more chlorophyll, then you're going to absorb more light energy, so you're going to be able to have an increased photosynthesis rate. So these are limiting factors. So what I mean by limiting factors, this is not just one of those things where, oh, if I increase the amount of carbon dioxide, the more I have it, it's going to keep increasing on a graph. It's not going to be a straight line up. You see on the right there, it shows that it actually plateaus at a certain point. When the graph increases, so as you can see in the beginning, as carbon dioxide concentration increases, the rate of photosynthesis also increases slowly. However, once it gets to a certain point, it's going to plateau. It's going to level off. And the reason is because something else is limiting it. So that means carbon dioxide is fully enough that's not affecting the photosynthesis anymore. There's a lot of carbon dioxide, but things like maybe temperature or light intensity is not enough. And that's when it's the rate of photosynthesis no longer changes unless, let's say, the light intensity starts to increase afterwards. So that's kind of the understanding of it. It is a bit confusing if you have questions to ask. There is also on the graph showing you the difference between a cold and a hot day. So this is what I mean. You can see on a cold day, it's going to plateau a lot earlier than a hot day. You can see on this point, this is where it starts plateauing. Up to, let's say, like 20% of carbon dioxide is already going to stop having an effect. On a hotter day, temperature, if it increases, there's going to be a higher temperature. You can see that it plateaus a lot later because the temperature is not as high of a limiting factor. But then again, it starts to plateau afterwards, maybe something like light intensity or chlorophyll is affecting it. So when you are answering a question on limiting factors and they give you a graph and you see the photosynthesis graph where it increases and then levels off, immediately I will think limiting factors. There's something limiting it that's causing it to level off. That will be my first thought. This is a massive question. Like, it's very big, but all of these are very self-explanatory and easy. So I'm just going to work through these with you. First part is asking you to describe how energy for the photosynthesis reaction is gained by plants. I see that the point is 2 marks, so I need to write 2 points. First thing, how energy for the photosynthesis reaction is gained by plants. It's going to be light. That's the only thing that could be. And how is this light absorbed? How is this gained by plants? Light is then absorbed by chlorophyll, or you can say chloroplast. So that's my 2 marks. Second thing, calculate mean value of X. This is just a math question. Add the 3 values together, divide them by 3. That one I don't really need to explain much. Again, if you're struggling with math questions, do as many as you can, but they should be given marks. They should be really easy marks. So the third one, draw a ring around the anomalous result. Now in this question, when they ask you for anomalous results, the only thing you need to look at really is what is sticking out. What doesn't match the others? So let's have a look. 20 looks pretty similar. 25 looks pretty similar. Same with 30, same with 35, same with 40. When we get to 45, we see it's 1.9, 2.2, and 14.2. So I immediately think that is the anomaly. That doesn't make any sense. It doesn't match any of these values. I'm going to circle 14.2. If you still don't know what the anomalous result is and they give you a table with the mean as well, you can figure this out by seeing if the mean matches. So in the question, 1.9 plus 2.2 divided by 2 is 2.1. It obviously didn't include 14.2. That's an anomaly. That's one way to figure out. So if we want to look at that, we can also point out that when you're doing your own like graph questions, just be careful as well to know that if there's an anomaly, you do not include it in your calculations. This is something to really note down because a lot of students may get this wrong. They see an anomaly, they still include it in their mean. Do not include it in their mean. So in the question before, it asked you to calculate the mean value of X. Let's say, for example, test 1 was anomaly, you ignore that result and you do the mean of the other 2. That's something to point out. Next question is asking you give one factor that the student should have kept constant in this investigation. 1 marker, also pretty straightforward. And in this question, it's asking to investigate the effect of temperature. So basically, any answer other than temperature is going to get you a mark. So just quickly showing you the answers for that, you can see here from 1.7. Light intensity, carbon dioxide, pondweed size amount, pondweed species, same pieces of pondweed, like distance of it. All of this is going to just get you the mark as long as you don't answer temperature. Just do not answer the independent variable. So what they're investigating in your answer, if they're asking you, what are they going to control? Any control variables, anything but the independent variable is likely going to be the answer. This question, let's have a look. Why did the rate of photosynthesis decrease from 35 to 45? So this question we also had a look at earlier. As I mentioned, when the temperature increases to way too high, your enzymes could denature. And because they denature, they can no longer bind to any molecules, no more substrate, blah, 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 all of that doesn't work, stops working, no more photosynthesis. So the answer to this question is generally just enzymes denature, their active site changes shape, so photosynthesis decreases. That's kind of more thinking a bit further, although this is a slightly harder 1 marker. With 1.9, this is just a quick, I kind of just want to go through quickly on graph skills. It tells you to label the y-axis using a suitable scale, plot the mean data, draw a line of best fit. So when you're drawing a line of best fit, just make sure that it is linking all the lines and it's one straight line, like not straight line, but it's all just like you're drawing it with one stroke. You're not doing like ruler, one point to the next and then one point to the next and then one point to the next. One line, just linking all of them. That's the line of best fit that you want to draw. So just be very, very careful when you're drawing graphs. And this is one of those things, again, same with math. Do as many practice questions as you can because a graph question will always show up in a biology exam. So make sure you can grasp the skill of answering the graph questions. So that will be kind of the answer. Here it tells you just the enzyme loses its shape of the active site, starts to nature. That's the answer. Correct line of best fit. There we go. Let's see. I saw a question earlier, so just want to look at that. Lunch break about 12:30, 12:45. It's kind of like, yeah, the midpoint between about 2.5 hours in, so about 12:30, 12:45. Okay. So now let's look at respiration. So this is something that we want to look at. Again, this is very important. Respiration is a topic that shows up almost every exam, so let's have a look. Aerobic respiration, or just respiration in general, is an exothermic reaction. So when I said endothermic and endothermic is when you take in energy and exothermic is when you release energy. Aerobic respiration is an exothermic reaction because it releases energy. So aerobic respiration, extremely important because it continuously occurs in living cells and it allows cells to live because it provides continuous energy. And we need to know the difference between aerobic respiration and anaerobic respiration. So with aerobic respiration, it uses oxygen and completely is the keyword, completely oxidizes it. So the simple equation we have is basically the exact opposite of photosynthesis. C6H12O6 plus 6O2 gives you 6 carbon dioxide and 6 water, H2O. Anaerobic respiration takes place without oxygen. So it's incomplete oxidation of glucose. So you're going to have to highlight that word. Aerobic is completely oxidizing it using oxygen. Anaerobic is incomplete oxidation without oxygen. There are 2 types of anaerobic respirations you need to know, and that's one in the humans, in our muscles, and one is in plants and yeast cells. In the ones in our muscles is when glucose is converted to lactic acid. Now, this is good for applying questions. So there was a question earlier about the heart cell, the heart, like the coronary heart disease. This is where it kind of applies a little bit. With anaerobic respiration, you produce lactic acid. And whenever, I don't know if you guys feel this, but when you're doing exercise, sometimes your muscles may feel sore. This is the reason. Your muscles will feel sore because you're producing lactic acid. You're running out of oxygen, so your body's relying on anaerobic respiration. So then you produce lactic acid, this goes into your muscles, and you're going to start to feel muscle fatigue, causing your muscles to feel sore. That's kind of like how, it's kind of cool, I think, kind of cool. Anaerobic respiration in plants and yeast cells, the glucose is converted to ethanol and carbon dioxide. And this specific example you need to know where the anaerobic respiration in yeast cells, we use this process called fermentation. So this ethanol and carbon dioxide produced usually is for things like alcohol or bread. So you do need to know that anaerobic respiration in yeast cells, you need to know it's called fermentation, and you need to know that they produce things like alcohol and bread. So the ethanol produced is basically alcohol that adults drink. So that is that. One thing to also point out, the aerobic respiration in eukaryotes, they do take place in the mitochondria. Sometimes they do ask you where it does take place, so that's something to keep in mind. It is in the mitochondria. But this is aerobic respiration, and most of it. And this is only in eukaryotes, just to, eukaryotes, aerobic respiration, it's in the mitochondria. That's just something to point out. Okay, right. This is a really good question that involves both photosynthesis and respiration. But before are we gonna go through cell division such as mitosis and meiosis? I don't think so, because I think that is mainly triple sciences, and this one is obviously both combined and triple. However, if you have any questions on this topic, feel free to ask and I can try and answer it at the end of the biology session. So yeah, if you have any questions on mitosis and meiosis, ask away in the chat and I will answer them in a bit, but we'll focus on respiration now. So here is an exam question. I'll give you guys maybe a minute, or I'll give you guys like 2 minutes to look at this question, try and answer it, and then I'll go through it. Okay, let's have a go at just explaining it very quickly. So first question is asking you why the concentration of carbon dioxide in the tube stays the same between day 0 and day 5. Based on the question, we know there's pondweed and a pond snail in the boiling tube. So the pond snail is respiration. The pondweed is also respiring, but it is also carrying out photosynthesis. What do we know about respiration and photosynthesis? We know that respiration, we're going to be using, we're going to be taking in oxygen and we're going to be releasing carbon dioxide. Photosynthesis is taking in carbon dioxide at the same time. So that explains why there's no change, because for photosynthesis in the pondweed is taking in carbon dioxide, and the pond snail and the pondweed also respiring, releasing carbon dioxide. So this causes no change because they're releasing and taking in at the same time. So that's kind of the explanation for that. 2 marks. Second thing, suggest why the concentration of the carbon dioxide increased between day 5 and 10. So this is what I meant by like suggest question. It's not really like your like topic knowledge, but more based off reading off the graph. So it's asking you why concentration has increased. What do you know about between day 5 and 10? It tells you after 5 days, the scientists completely covered the boiling tube with black paper. When you completely cover the boiling tube with black paper, no light's going in. That's the first thing I can think of. No light, absolutely no light, because it's black paper and there's nothing going in. If there's no light, what process doesn't happen? Photosynthesis. So my thought is, okay, if there's no photosynthesis, no carbon dioxide is being taken in, and therefore it's only carbon dioxide being released through respiration. Therefore, there's an increase. That is the second question. Third question This one I'm just going to explain to you because this was not covered earlier, but it's asking you to explain the death of the pond snail caused by the concentration of carbon dioxide to increase after day 10. I believe this is only, I'm unsure about this. I think this is mainly triple science, but I'm unsure. Don't take my word for it. But here it tells you when this is when a pond snail dies after day 10. What happens when an organism dies? Essentially, it decays. It gets broken down, it gets decomposed. And this is being broken down by things like bacteria or things like decomposers. So when these are being broken down, these decomposers of bacteria do respire as they're being broken down, therefore releasing a bunch of carbon dioxide. So the answer to this question is the snail dies. It is being decayed or being decomposed by these decomposers. And therefore, these decomposers respire. They respire, releasing carbon dioxide, and therefore causes the increase in carbon dioxide. That is the way to explain it. But the extra information that I basically just told you is that when an organism dies, it gets decayed and broken down by these decomposers and bacteria, and they respire. So here are the answers. Again, you will have these slides so you can have a look at that. But it covers mainly photosynthesis and respiration to a T. I don't think there's much more outside of this. Any questions regarding photosynthesis and respiration? Please type it in the chat. If not, we're going to move on. Okay. Right, now let's compare the nervous system and the endocrine system. So this is I really should look at the questions really quickly. So the nervous system and the endocrine system comparison, there are similarities and differences. We'll just have a quick look at that. The nervous system, and endocrine system, the similarities is that they're both involved in homeostasis. You're going to see that in homeostasis. And the changes in external or internal environment of these organisms, they act as stimuli. So if there's any change in the environment or what's happening inside your body, this is going to be acted as a stimuli or a stimulus. And the nervous and hormonal system, so endocrine system, another word is the hormonal system, they coordinate a pseudo response to this stimuli. So any changes in environment or any changes in your internal environment is going to produce a suitable response. The difference is what they're made up of, the signals, and how it all works. The nervous system is made up of nerve cells, whereas the endocrine system is made out of glands. Nervous systems, the signals are electrical impulses, and they travel along the nerve cells through synapses. The endocrine systems is chemical signaling, so it's through the blood circulation. The nervous system is very, very quick. It happens in seconds. So you moving or me talking, any type of coordination is going to be electrical. It's going to be very, very, very quick. The endocrine system, however, is a lot slower. It usually takes minutes or even hours for it to work. However, the duration of effect, they are very short. So the signaling stops once the neuron stops signaling. Once it stops signaling, there's no more signals. It's very, very short. The endocrine system, on the other hand, is very, very long. It continues up till basically the hormones stop being produced. So it could be years. So the difference, yes, it is very fast for the nervous system, but it's very short-lived. Endocrine system is a lot slower, but it's a lot longer, lasts a lot longer. So again, remember when you're comparing these, you talk about both similarities and differences. Don't just talk about differences. This is a very common question that comes up. This is very, I'm not going to explain this much. I think go through this question yourself. I'm not going to go through this question because generally this one's more like applying your knowledge. But yeah, have a look at this question. It's a really good exam question. The one I really want to talk about is homeostasis, water balance, because this one is the one that students get confused with a lot and because it's so complex. So I'm going to try and break this down as slowly for you as I can. So with water balance, if there is too much or too little water in your body, this can cause your organs to not function very properly. That's why our body needs to be able to coordinate the level of water in our body. So if our body fluids are too dilute, so if there's too much water, this goes all the way back to our transport topic, osmosis. If there's too much water in our body, water is going to go into our cells through osmosis and causing the cells to possibly burst. The opposite might happen if there's too little water in our body. There's going to be more water in the cells, water is going to move out of it through osmosis, and the cells could shrink. Therefore, it's so important that we coordinate any the water balance. So removing any excess water, ions and urea, and this all takes place within the kidneys. This produces urine and maintains water balance. So sometimes you might think, why is my urine more clear on other days and why is it more yellow on other days? It's because there's either more water or less water in your urine, depending on how much water you have in your system. So this water balance is controlled by the hormone ADH. If you want to know the full name of ADH, it is antidiuretic hormone. I'm gonna type that in the chat just in case anyone wants to know. Because sometimes they do give you the full thing. Diuretic. Let's spell this wrong. Antidiuretic hormone, that is ADH. And this is released by the pituitary gland when the blood is too concentrated. So when there's too little water in the blood, that is detected, and the ADH is going to be released by this pituitary gland once there's too little water in the blood. The pituitary gland is essentially the master gland in the brain. It secretes multiple different hormones and in response to multiple different body conditions, but the one you mainly need to know is ADH within water balance. The role of ADH is to increase the permeability of the kidney tubules. So if it's more permeable in the tubules, more water is going to be reabsorbed into our body, into the blood. And this is a very classic example of a negative feedback and controlled. So if the blood is more concentrated, so there's very little water in the blood, there's going to be more ADH released. So that's negative feedback. So I'm going to explain the concept of ADH again because it can be confusing. With the release of ADH, this causes our kidney tubules to be more permeable, meaning more water is going to be able to be reabsorbed. And this is going to be confusing because of the next question I'm about to show you. Here is a question on ADH. I'm going to give you guys a minute to look through this and I'm going to explain it. This is where it gets a bit confusing, but I'll give you guys a minute to. Okay, hopefully you have read the question. So it's asking you how the decrease in concentration of ADH would cause the changes shown. And the decrease, it shows you that the concentration of dissolved substance in the urine is very low, and the rate of urine production is very high with a low concentration of ADH. So now I'm going to go through why this is. As I've said, ADH makes the kidney tubules a lot more permeable, meaning more water is going to be able to be reabsorbed. In this case, talking about decrease. So if there's less ADH, it's going to be less permeable to water. Less water is going to be reabsorbed. If there's less water reabsorbed, more water is going to be in your bladder. So this is the confusing part. If there's water reabsorbed into your body, then there's going to be more water in your blood, and you're going to have less water in your urine, in your bladder. However, if there is less ADH, less permeable kidney tubules, less water is being reabsorbed into the blood. Therefore, more water is going to be in your urine. If there's more water in your urine, you're going to want to have to go to the bathroom more. So rate of urine production is going to be a lot higher, explaining the higher urine production in the table. Concentration of dissolved substances in the urine is a lot lower because there's more water. So in contrast, in concentration, it's going to be a lot lower. So the answer right here tells you here, tubules are less permeable to water, less water is reabsorbed, more water in urine causing increased rate of production, and more water in urine causing lower concentration of dissolved substances. So that is water balance. Anybody have any questions regarding that? Because it is very wordy. If not, I am going to move on to the next part of nervous system and homeostasis and all of that. So the nervous system. The nervous system, you need to know a few things. The nervous system allows humans to react to their surroundings. The nervous system essentially coordinates all of your behavior, all of your movements, like even your eyes, your smell, your talking, your taste. Everything is coordinated by the nervous system. So it's very, very important. Sorry, someone asked me to explain something again. Which part do you want me to explain? Can you please elaborate? Because is it the ADH stuff? Because I know that is quite difficult to understand. Yeah, could you elaborate on what I'm explaining? At ADH. Okay, yeah. It is a hard part. Don't worry. I'm gonna go back to it. We'll take our time. Hold on. Okay. Right. With ADH. So think of okay, I have a cup right here. Think of this. This is my matcha. Think of the if you guys can see my camera, this will be very helpful because it's better to visualize it. Think of this as your kidney tubules and everything surrounding it like is just like water, whatever. This is one where you want to put in your blood. Your blood has very low water right now. You want more water in your blood if it's too low. ADH releases and causes these kidney tubules to be more permeable. More permeable, meaning more water can go in. I hope that makes sense. If it's more permeable, more water can go in. If there's more water being absorbed back into the blood, in this case, then more water is going to be in your blood, and there's going to be less water when you go to the bathroom, when you pee. And the reasoning is because when you're reabsorbing all of this water into your blood, it's all going to be in your blood. It's not going to be left to like leave your body. Once it's in your blood, it's in your blood. You're not going to be, unless you're sick, you're not going to be peeing blood out. So when there's ADH released, tubules become more permeable. More permeable, more water goes into these tubules and into the blood. Once water has entered the blood, then there's going to be less water left to go to the bathroom or whatever, left basically outside of blood to release when you go to the bathroom and the urine, all of that. So more ADH, more permeable, more water reabsorption, less in the urine. If there's less ADH, less permeable, less water going into blood, more water left in the rest of the body to be released. If it's anything that's confusing about what I said, is everyone okay with what I said? It's okay if you want me to explain it again, what exactly was confusing about that. But if is there any questions on that? Like, is everyone good with that? I'm hoping everyone is, because it is quite confusing, but okay, great. You can understand now. Great. Is AQA for triple or combined Q and GCSE? I think ADH is in higher tier, and I think it might be in combined. I'm gonna double check for you. Let me just have a look at my list. Give me a second. Hold on. Where is it? Okay. I think, yes, I do think that ADH is probably in combined. Yes, ADH is in combined, unfortunately. So you're gonna have to understand that, yeah. Nephrons. Okay, that one is also very, very specific and also only in, I think, triple and higher, and also only in AQA, if I'm not 100% sure on that. But I don't think we're going to go through that unfortunately today. But if you are if there's anything confusing about nephrons you want to know about, again, you can ask, because I know someone asked about mitosis and meiosis. So if there's a specific question you want me to go through in nephrons, I will answer that. So put the question in the Q&A box and I will do that after the biology session. Okay, let's move on to nervous system. As I've said, coordinates your behavior, information passed along the neuron cells, and their electric impulses, as mentioned before. These electric impulses go to your CNS, the central nervous system, your CNS being your brain and your spinal cord, but we're talking mainly the brain. Let's not confuse ourselves. The CNS, as I said here, brain and spinal cord, and it coordinates responses involving effectors. Effectors can be things like your muscle. That's an effector. So there are 3 types of neurons you need to know about. There is the sensory, the relay, and there's also the motor. The sensory, relay, and motor, it's kind of think of it as like a linking from A to B to C. It goes from sensory and then to relay and then to motor. But the steps regarding it is you have receptors, so things that detect the stimulus. So if there's a change in environment, receptors are going to detect it. So for example, touch. There's going to be receptors on your fingertips. You're going to feel this touch. Receptors detect the stimulus, carries the electrical impulses through the sensory neurons from the receptors to the CNS. This electrical impulse is going to go through the sensory neuron. Then it's going to go through the relay neuron, through the CNS. And after you've gone through the CNS, through the relay neuron, this electrical impulse is going to continue down to the motor neuron and from the CNS, and this is going to be linked to the effector. The effector is then going to produce a response. And I'm going to show you an exam question later to show you kind of what I just said. But the steps essentially is just receptor, sensory neuron, relay neuron, which is through the CNS, motor neuron, effector, response. Step by step. I'm going to say that again because that's usually the easiest way people understand it. Response. So, no, sorry, not response. Sorry. Stimulus, sensory neuron, relay neuron through the CNS. So CNS, yeah. And then motor neuron, effector, response. That is step by step what is exactly is happening. So I'm going to show you an exam question on that. Okay. Oh, there is no exam. Wait, okay, where is it? Let me just go all the way back because I think this is a really good example here. So I did say we won't go through this, right? So it asks you to describe how the diagram brings about this reflex action when someone touches a hot pan. So when someone touches a hot pan, like you're going to detect this temperature. It's hot, so you're going to want to move this away. So when you detect it, receptor detects the stimulus. So the stimulus being the heat. So you see the first one here, stimulus, heat, detected by temperature receptors in the skin. This electric impulse is going to then travel through the sensory neuron to the CNS. This is then going through the CNS, through relay neuron, and then to the effector through the motor neuron, so motor neuron and then effector, and then the muscle contracts, which is the response. So you have to be more specific when you're answering question through the response. Also, there's another point you're talking about chemical transmissions across the synapse. Don't stress too much about that point, but it's just something you need to know when you're answering the question. As I've said, between neurons, there are synapses, and this is how they link to each other. They have signal to each other. Between these synapses and these neurons, the transmission of signals is through chemical transmission. So that's just something to point out. But that's kind of the whole of the nervous system. If you have any questions on just the basics of it, please let me know. The next section for the eye is only in triple science. So if you're in double science, I guess have a break. Like in combined science, have a break, but the next part is triple science. So I'll just quickly explain this. For triple sciences, you need to know about the eye very specifically. So there are 2 things that you need to know about the accommodation of the eye. First one is looking at just different distances, looking at something close and looking at something far away. Then for adaptation to light is looking something very bright compared to looking something very dark, very dim light. So let's look at that. You need to know the structure of the eye, first of all. I'm not going to go through every single structure and every single function. It's in the table right there if you want to have a look, but you do need to know them well. So if you're looking at something near, and if you look at different distances, we have our ciliary muscles and our suspensory ligaments, and they work antagonistically. What I mean by antagonistically is that they work together, but opposite. You'll understand what I mean when I explain that. If you're looking at something near, your ciliary muscles are going to contract, your suspensory ligaments are going to loosen, and the lens becomes shorter and thicker to see something near. So you can see how they work oppositely. Ciliary muscles contract, suspensory ligaments loosen, but they work together to allow the lens to become shorter and thicker. This allows us to see things near. Things that are far away, opposite. Ciliary muscles relax, suspensory ligaments are pulled tight, so contract essentially, and the lens is thinner and longer. So that's looking at things at different distances. Adaptation to light is when there's different light intensities, and this allows our pupils to dilate and constrict. So the reason is because if there is a very bright light, you don't want to damage your eye, so it constricts, makes it smaller, so not too much light is going in. And if it's very dim light, you're going to have these your pupil dilate, so that when it's larger, you get more light in, so you can see in darker light. So in this case, when there is a very low light intensity, the radial muscles contract and your circular muscles relax. Again, they work antagonistically to allow the pupil to dilate or constrict. And in bright light is the opposite. Radial muscles relax, circular muscles contract. So that's the eye. There are a few questions on here. However, I am wary that there are people who are doing combined sciences here as well. So if you are doing triple sciences, have a look at this after the seminar, after the, oh my God, hold on. Right. The light's off again. But yeah, if you guys have any questions on the eye, just look through it yourself after the thing. Okay, can you go back to the eye? Yes. What do you want me to look through in the eye more specifically? Yes. Okay. Yeah, no worries. Just wait, hold on. Give me a second, guys. The light is off. Okay, sorry. Okay. Perfect example for light and dark right now for the eye. Okay, let's explain that. So I'm going to explain it again with the circular muscles, radial muscles stuff. Okay. I think everyone, kind of understood the near and far one. I feel like that one was, I think, explained more clearly than what I said to the light thing. So let's go back to the light. So when you're in very, very bright light, your eye is going to want to, your pupil, so basically the hole that allows light into your eye, is going to want to go smaller, constrict. They're going to want to go smaller because you don't want to damage the eye. Too much light in the eye is going to damage it, yeah? So they're going to constrict the pupil. When they constrict the pupil, less light is going in. And how they do this is by relaxing the radial muscles and contracting the circular muscles. If there's dim light, so it's very dark, you want more light to enter your eyes because you want to be able to see. You want to be able to figure out what's going on in the dark. In this case, your radial muscles are going to contract and your circular muscles are going to relax. And this causes your pupil to then dilate. So it works very similar to the distances where if it's closer, they work antagonistically where one contracts and one loosens. This is the same thing. One contracts, one relaxes. And it allows the changes of the pupil to become bigger or smaller, so dilate or constrict, depending on the light environment. And the reasoning behind this is because if there's too much light, you don't want to damage the eye, so you make the pupil smaller. And if there's very little light, you want the pupil to be bigger so that more light can go in so that you can see more clearly. Was that clear enough? Is everyone in triple science understanding that better? Which are radial muscles? I don't think you need to know where they are. I don't think they ever ask you that. So hopefully that was clear enough. Yeah, perfect. Yeah, okay, let's move on. Okay. Okay. Now, I want to give you guys, this is not on the eye. Actually, this is on reaction time, and I believe this is in both triple and combined sciences. So I want everyone to kind of have a look at this question. It's about reaction time, and I want to kind of look through this because planning and investigation question, I didn't talk about this in command words, but planning and investigation questions are very, very common in biology. And they're very, very useful because there's a very specific structure to how they want you to answer it. So I'll give you guys a minute to look at this question and I'll go through it. Okay, hopefully everyone has had a look at that question. The way you want to do plan and investigation questions, I know this topic might be a bit harder for combined sciences, but the reason I'm doing this is because plan and investigation questions come up in every single exam, and the structure is going to work for every single person exactly the same way. So let's go through it. This one is just talking about reaction time. Rather than me talking about the answer, I just want to talk about how you formulate the answer. With planning and investigation, the first thing you talk about is just basically describing the experiment you're doing. So in this case, probably the ruler experiment, like dropping a ruler and then catching it. I think some of you had done this before. And in this case, just describing what's happening in the experiment. Drop the ruler, hold it, and then you talk about the variables. You talk about what you need to control. Sorry, let's talk about independent variables. So what you need to change, what you're going to do with the experiment, who's taking the coffee, who's not, how much are you giving. Then it's the control variables. This is another section where they want you to get marks from. So what you would control, the amount of coffee you're giving them, the type of coffee you're giving them. So to answer the plan and investigation question, you would talk about the experiment. So you're describing the experiment. Then you're going to talk about the controlled variables. And the last mark is results. So it could either be analyzing the results or making sure the results are accurate. So things like repeating the experiment or writing a table to analyze these results. Anything along those lines will be fine, and write 2 to 3 points in each section. Write 2 to 3 points of describing, 2 to 3 controlled variables, and 2 to 3 points explaining the results and how you'd analyze it. So here is the mark scheme. So you can see here, the top part tells you describing the experiment, middle part talks about explaining the results and how you make them accurate. So doing it multiple times, comparing results. Final thing, control variables. I hope this is clear for everyone. This is just a method to answering exam questions. Okay, again, this is going to be heavily on triple sciences. Combined sciences, you do need to know base level of all of these, but you don't need to know it in too much detail. But I'm still going to go through in more detail than combined sciences need to, and I'm going to go through in detail for the triple sciences. Okay. DNA structure. This one, combined sciences, you need to know. It's a long polymer. It's made of repeated nucleotide units. And the nucleotide units, you need to know the structure. You need to know what each of those things are. So you see on the right, the little structure there, it shows you nucleotide with the pentose sugar. You need to know it. The pentose sugar, the pentagon, yeah. The base is the rectangle. The circle will be the phosphate. And you need to be able to label all three. That is what the nucleotide is made of. The base can be either A, T, C, or G. You do not need to know the specific names of A, T, C, and G. If you're curious, I can tell you, but you don't need to know it. You only need to know it in A level. So they're always complementary, these bases. So A is always going to be with T, C is always going to be with G. And when I talk about this DNA and A, T, C, G, this is referring to replication and all of that. Okay. You see on the structure there? The DNA is also a double helix, we know that. And then within the DNA, there's a section of the DNA is called gene. Within the DNA, there are chromosomes, and the chromosomes are in a cell. So sometimes they can ask you about like which one's bigger, which one's smaller, blah, blah, blah. It starts from the time, I'm going to start from the smallest. It goes from cell to chromosome to gene to DNA. That is my way of explaining. Sorry, that was wrong. It goes from, not from large to small, sorry. It goes from gene DNA within the chromosome in the cells. Sorry, that was a bit of mix up. That's what I'm largest to smallest. Cell, chromosome, DNA. So you can see the diagram on the right. It shows you like in the cell, there's a bunch of chromosomes. In the chromosomes, there's a bunch of DNA. And then DNA, there's sections of genes. Okay. This is more on triple. Between the nucleotides, they have phosphodiester bonds. Between the bases, they have hydrogen bonds. Some exam boards need to know that, some don't. Transcription, you need to know it takes place in the nucleus. And the mRNA molecule is formed from a section of DNA that codes for a particular protein, so a gene. And translation, it takes place in the ribosomes. So transcription is in the nucleus, translation in the ribosomes. So this is the details that you need to know. The tRNA carrier molecule, they will have these amino acids like bound, they have this amino acid attached to these tRNA that are specific to the mRNA. So there's every 3 bases on the mRNA that code for a specific amino acid. 3 bases, so let's say ACC, codes for 1 amino acid. And that tRNA is going to bring that specific amino acid to the triplet codon, or the 3 bases, basically. Then it's going to keep going, as you see on that diagram, it's going to keep going. And then it's going to complete the chain, and they're going to form a protein. Between these amino acids, they're going to form peptide bonds, but they're going to form this protein. Then they start to fold, and then they fold, and they become either an enzyme or a hormone that is helpful to the body. So this is how translation works. I know I went very briefly on transcription. Please do more studying than what I'm explaining to you now, because it's in a bit more detail. But translation that's kind of the gist of it. Okay. I don't think I'm going to go through these questions. Again, look through this yourself. Nutrient cycles, I want to talk about the investigations of decay. This is also heavily on triple sciences. I do apologize for combined sciences, but this is also heavy on triple. Decomposition, something you need to know. It's a breakdown of large organic molecules to smaller molecules. So we did talk about this briefly earlier in the photosynthesis and respiration topic, but I'm just going to go in a bit more detail now that we are going through nutrient cycles. So decomposition, usually happens when a organism dies and they're decomposed. So they're being broken down. And what exactly on a molecular level is happening is that they're breaking it from a large molecule to a small molecule. And this is carried out by things like bacteria, fungi, or decomposers. There are multiple factors that affect this. So there's temperature, oxygen, and water availability. All of these, the more that the increasing in temperature, the more oxygen, the more water there is, the increase the rate of decay of these biological material, because the bacteria and fungi are going to be able to grow a lot quicker with all of these things. So to break it down a little bit more in detail, the increase in temperature allows there to be more kinetic energy, and therefore there's going to be a higher rate of decay. Similar to any other process, if the temperature is too high, enzymes are going to denature, and then it's no longer going to work. Water availability, if there is a increase in water availability, there's going to be a higher rate in decay, because water is required for decomposers, the digest enzymes to function. You need water for these enzymes to work. Therefore, more water is going to be better for decay. Oxygen, it's good generally, but this one's be careful with this, because it's only good for aerobic organisms and they inhibit anaerobic organisms. So if they're anaerobic organisms, meaning they don't rely on oxygen, a lot of oxygen is not going to be useful for them. I feel that feels very, like in the name. Anaerobic if there's oxygen, is not going to be helpful. It's the opposite. If it's anaerobic and there's no oxygen, obviously that's going to be better for those. The rate and chain of decay is just change in value over time. You might get a math question on that. And with gardeners and farmers who are trying to make these decomposition happen, usually they find the optimum conditions for very, very quick decay. So a good temperature that's fast enough but won't denature, enough water, and enough oxygen. Because they want this decay to allow for these biological material to produce compost. And compost is a very, very useful material for gardeners and farmers to use. Anaerobic decay, again, I said it does not use oxygen, and this produces methane gas. And this methane gas can be carried out in this production can be carried out in a biogas generator, and this allows it to produce fuel. This is something, good to know. The most common practical that they use for investigation of decay is the milk experiment, and is when the milk decays. This is very important to know, actually. When milk decays, milk has lipids in it, so fats is one way to explain it. These lipids are going to break down into glycerol and fatty acids. And because fatty acids they're acids, they're going to make the pH very low because they're very acidic. And the way this experiment works is that they monitor the pH to figure out how fast the milk is decaying, because the faster the milk decays, the more it's going to turn into glycerol and fatty acids. The more fatty acids there are, the lower the pH. So they usually they set this up with different temperatures, different pHs, and they do it at different intervals. So we're going to look at this experiment right now with one of these questions. So here, this is a math question. Again, look through this. We're not going to go through this. On 4.5, it tells you milk contains lipids, and lipids are broken down when milk decays. And it's asking you to explain why the pH changes more quickly when the temperature is higher. We know that as temperature increases, the rate of decay increases because there's more kinetic energy. If the rate of decay increases, the lipids are going to be broken down more quickly, and therefore more fatty acids are produced. Therefore, the pH is going to change much more quickly, and the pH is going to drop down so much more quickly because there's more fatty acids being produced. I hope what I've said is clear. Here are the answers. Enzymes are more active, more bacteria produced, lipids are broken down more quickly, and therefore fatty acids produced more quickly. Any questions on everything we've done so far? Any questions? Any questions on that? Okay, can you explain control variables, independent and dependent? Yes. Control variables is what you want to make sure it's fixed, so you make the thing fair. So if I were trying to figure out how temperature affects let's say, affects respiration, let's not do respiration. Let's do that. How temperature affects respiration, or not, photosynthesis. Let's do photosynthesis. How temperature affects photosynthesis, sorry. The control variables would be anything but basically temperature, because the independent variable, in this case, the independent variable, which is the thing you're investigating, is the temperature. That's the independent variable. Control variables are things you don't want to change. Things like the number of plants, the type of plants, the light intensity in the room, the oxygen, any variable that could affect the experiment, control it to make sure it stays the same for everything. Anything but the independent variable, to me, is control. Dependent variable is what you're hoping to find. It's like your hypothesis. So in this case, the dependent variable would be, you think that an increase in temperature is going to allow for a higher rate of photosynthesis. Is that somewhat clear? I can try and explain it again, but that was my take at answering that question. Okay,